Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Jae Sung. Dr. Sung is Assistant Professor in the Microbiome Program at the Mayo Clinic Center for Individualized Medicine in Rochester, Minnesota. Dr. Sung's research aims to provide insight into health status from a gut microbiome sample. He introduces the Gut Microbiome Health Index, a biologically interpretable mathematical formula for predicting the likelihood of disease independent of the clinical diagnosis. Today, Dr. Sung presents his session, A Predictive Index for Health Status Using Species-Level Gut Microbiome Profiling. Dr. Sung, over to you.
Hi, everyone. Thanks for joining us, and it's a pleasure to be speaking to you all. My name is Jae Sung, and I'm an assistant professor at Mayo Clinic in Rochester, Minnesota. Today, I'll be presenting one piece of work from my research group in understanding more about what the gut microbiome can tell us about our health. So as many of you may be aware, we're now in an era where the clinical practice is co-evolving with the accelerating presence of large-scale data sets. On the left are probably data types that almost everyone here knows about, and on the right are high-throughput data sets of not only biological measurements, such as genomes, transcriptomes, and microbiomes, but also imaging data, wearables like your Apple Watch or Fitbit, social network data, and also mobility data. Despite their enormous potential in advancing precision medicine in the clinic, the problem today is that they can be tremendously hard to discern signal from noise and also difficult to interpret from a biological mechanistic point of view. In the clinical setting, if the pace of how much we're generating large-scale data sets of cells, tissues, and entire organs, and soon to be entire bodies is only accelerating, then it's not that difficult to imagine that the patient of the future will be completely digital and virtual. Basically, without having to walk into the clinic, a patient can send his or her digital twin composed of dense, dynamic, and personalized data clouds to then receive predictions regarding diagnosis, prognosis, and therapy selection and health optimization. Now, as pretty much everyone here in the audience knows, the human microbiome and gut microbiome in particular has a strong influence on almost every aspect of human biology. Here on the left, I've provided a few examples where the gut microbiome has been shown to play a significant role in human physiology. It's widely considered that when this population of microorganisms is disrupted, thereby leading to certain pathways and functional modules going wrong, then the result can lead to an assortment of diseases, such as the one listed on the right. Now, as many laboratories around the world are trying to figure out specific mechanisms of how microbes lead to disease, or how diets and drugs modulate the gut microbiome, or how to develop bugs as drugs, we are, on the other hand, interested in how to apply microbiome data, or in other words, shotgun metagenomic or 16S gene sequencing data with phylogenetic tree information and with biochemical gene annotations, and to go beyond profiling taxonomies or functions and create online live dashboards, apps, and clinically impactful yet biologically interpretable knowledge that can help us make clear decisions for patients. For example, metagenomic data have been used to identify infectious pathogens on the surface of prosthetic joints, differentiate clinical subtypes of disease and understand subset-specific mechanisms, in this case, irritable bowel syndrome, be able to predict post-meal glycemic response, predict whether a patient with inflammatory bowel disease will respond to biologic therapy. And also in one of our group's most recent studies, by looking at the gut microbiome data of patients with rheumatoid arthritis, we're able to predict whether a patient will eventually show improvement in their symptoms at follow-up visit. This was a cool case of how the integration of high-throughput data sets, low-dimensional clinical data, and a deep learning neural network can be applied to answer clinically meaningful questions. But one question we didn't view as being properly addressed is the question of what are the specific traits of a healthy gut microbiome, and how do you measure that? But why is this important? Well, for starters, it can give you a destination point of what you want your gut microbiome to look like. You can talk about prebiotics and probiotics and FMT and the so-called microbiome diet all you want, but without knowing what to aim for, it's really hard to make much progress on a macro and population level scale. And just as importantly, you can use the gut microbiome as a barometer to see whether your health is in good shape, or some sort of, for example, some sort of a clinical index to tell you the likelihood of whether you're good or you have something bad brewing inside of you. And so we did that in a paper published in the fall of 2020, led by a great postdoc in the group, Dr. Vinod Gupta. Now, this paper is going to be the focus of what I'll talk about in the next 30 minutes. Now, as a bit of background, this study was motivated by an interesting meta-analysis that came out of Eric Alm's lab at MIT back in 2017, where they found that gut microbiome signatures can be shared across multiple studies and even multiple diseases. First, I'm showing on the left 3 heat maps, each corresponding to a particular disease, where rows are bacteria and columns are independent studies investigating the respective disease. In the case of obesity, colorectal cancer, and CDI, or Clostridium difficile infection. The darker the red means that the bacteria are higher in disease, and the darker the green means the bacteria are higher in controls. As you can see by comparing across columns, bacteria was either high or low in disease, often consistently across studies, showing some reasonable amount of reproducibility in disease-associated gut microbiome signatures. Excuse me, sorry. Okay, let's see here. But what was really interesting is what the authors found next. Bacteria whose abundance patterns were commonly observed across multiple diseases. On the heat map on the right, the columns are now bacteria, and the white rows with the bars indicate whether the bacteria was seen across 2 or more diseases, or towards the bottom half, unique to a single disease. As you can see on the top row, there are many bacteria that are consistent either up or down in disease across multiple diseases, hence non-specific to a particular disease. But towards the bottom, you see gut microbiome signatures found in only one disease. So there are indeed several bacteria that are disease-specific. Now, their observation that gut microbiome signatures can be shared across multiple pathologies led us to wonder the following. Are there gut microbiome taxonomies more likely to be observed in health than in disease? And if so, then how can we capture and harness that information to design a gut microbiome test that predicts health? Now, in our study, healthy was defined as self-reported to not having a disease or disease-related symptoms. Again, the operative definition of healthy in our study is no disease presence nor overt symptoms. Thus, our main aim is to design a stool-based test that predicts the likelihood of health, or how likely you are to have a disease or not. Now, this idea could be useful in the following. Say a patient walks into the clinic for a routine checkup or screening, gets his normal blood work done to look at fasting blood glucose levels, cholesterol, etc. And then integrated into this checkup would be a gut microbiome test, showing you not only what species or strains are up or down, but also any signs of something being potentially off. Actually, if you talk to clinicians, at least where I work at Mayo Clinic, they get asked all the time by their patients on how they can learn more about their gut microbiome and how it relates to overall health. So definitely a niche for this type of work exists. Okay, but where do we start? Now, this is a very simple infographic I bring up to my colleagues who aren't familiar with the biomarker discovery plus machine learning type of work our group does. Oftentimes people think that analyzing large-scale biomolecular so-called omics data entails a bunch of coding and data parsing. Although there are some elements of truth to that, rather, the majority of the time is actually spent on getting good quality training data, or the discovery cohort, necessary for the machine algorithm software to learn something new and useful. Having a little trouble with the mouse today. Okay, so afterwards of getting this training data, or essentially the search space we'll be working with, we can then focus our efforts on the experimenting and coding, which will involve feature selection, then model selection, and lastly, model optimization, or basically parameter tuning to fit the function to the data. So first, let me explain how we obtain the training data. Okay. Fortunately, all the data we need is actually available for free in public data repositories. Shown here in the slide is our pipeline on how we pulled together gut microbiome data from previously published studies. In short, we started from a collection of over 7,500 stool shotgun metagenome samples pulled from 55 independent publications. After excluding some studies and samples using a strict criteria, the final training data was a pooled metadata of 4,347. Let's see here. Okay, sorry. A pooled metadata of 4,347 stool metagenome or gut microbiome samples from 34 independently published studies ranging across 13 phenotypes. As shown in the table here, these 13 phenotypes include 2,636 samples from healthy subjects. And as I mentioned before, healthy is defined as self-reported to not having a disease or disease-related symptoms. Also, there were 1,711 samples from non-healthy subjects. Here, non-healthy subjects were actually patients with one of 12 different disease or abnormal body weight conditions. Now, you can say that we're looking at a very limited number of disease populations, but this was all we could find at the time. In that regard, something to point out is that, well, this is not true anymore, but at the time when the study came out, this was the largest metagenome study with respect to sample number for the gut microbiome. Anyway, these 12 non-healthy phenotypes included cardiovascular disease, colon cancer, Crohn's disease, ulcerative colitis, obesity, and type 2 diabetes. One thing to note here is that we actually downloaded all the microbiome samples as raw pre-processed sequence files and uniformly ran our in-house bioinformatics pipeline. Since we're pulling microbiome data from various studies, this uniform bioinformatics processing is really important for getting to get rid of as much batch effects as possible, although we acknowledge that it's possible to eliminate entirely. So again, this entire study was driven by publicly available data. So big thanks needs to go out to all the labs that generated and posted the microbiome data with no strings attached for big data scientists and computational biologists like us to do whatever we want with it. So this is to show what plotting 4,347 microbiome samples looks like once projected simultaneously in the two-dimensional space. In the principal coordinate analysis or PCOA plot on the left, we're showing all samples of healthy and non-healthy. And on the right, we're showing the exact same plot, but the samples are color-coded by the specific phenotype. Now, going back to the previous slide, across all of these samples, we found 785 microbial species observed to be present in at least one sample. Of note, our entire analysis was performed on the species level to get to the finest level of taxonomic precision while maintaining a reasonable amount of comprehensivity. Of course, we can look at much more granular strain level organisms, but tools for comprehensive strain detection aren't very reliable and credible yet. Believe me, we tested a variety of tools with our own in-house validation techniques. Okay, we decided to investigate the species a bit, mainly by plotting a histogram of prevalences of all these 785 species. Here, species prevalence is defined as the proportion of all 4,347 samples where that species is observed to exist. A prevalence of 100 means that the species is present in all samples. 50% means that the species is found only in half of the entire samples, and a prevalence of 0 means that the species is present in none. As you can see on the far left, the majority of species is present in a very small fraction of samples, and there is a skewed distribution towards the right. This means that the prevalence across samples varies widely depending on the microbial species. This is not really new news, and we're just basically confirming what others have previously found, but just on a much larger metagenomic data set. Looking into this further, we found that more than half of the 785 species were rarely discovered. And then we have over 300 species that are almost uniform in their prevalences. Interestingly, we found 6 microbial species that are prevalent in over 90% of samples. It's important to keep in mind that these 6 species are prevalent regardless of health or disease state. So we conjecture that these are possibly core species of the gut microbiome indicated to us by the actual data from thousands of microbiome samples. Okay, so that's a little bit about the training data we'll use to develop our gut microbiome health indicator. Next is to find which species are associated with health and which species are associated with disease. Okay. To explain how we'll do so, I'd like to use the following analogy. On one side of the screen, you have an environment that is teeming with life, fresh air, good overall ecosystem, but on the other, you have an environment full of waste and toxicity. Obviously, the healthier environment is the one on the left. If you think about it, in the good ecological environment, you have life forms that are much more prevalent than in the bad environment. But one important point is that should not be overlooked is that the bad ecosystem here on the right is not void of life, but on the contrary, there are definitely life forms in the bad environments that are much more abundant compared to the good environment, or in other words, scarcely seen in the good environment. So if we were to quantify this concept and adopt it for our own purposes, we can term the microbial species that are more frequently observed in healthy subjects as health prevalent species. We can also term the microbial species that are less frequently observed in healthy as health scarce species. So we have 2 important terms here, health prevalent and health scarce microbial species. Again, health prevalent species can be assumed to be associated with the healthy state, and these health scarce species can be assumed to be associated with the non-healthy state. So here's some data to further illustrate what I mean. We have a list of bacterial species, how often they're found in the 2,636 healthy samples, how often they're found in the 1,711 non-healthy samples or those having a disease, the difference between these 2 prevalencies, and the fold change. In the table at the top, we show the top 5 healthy associated species rank ordered based on how more often they're found in healthy relative to non-healthy. Some of you may recognize the Bifidobacterium adolescentis, which is a known probiotic. In the bottom table, we show the top 5 non-healthy associated species rank ordered based on how more often they're found in the non-healthy relative to healthy. You can see that at the top, they're mostly Clostridia and Ruminococcus species. So in summary, we have a prevalence-based strategy to identify species associated with a phenotype, which is basically about taking into account the frequency of the signal. Importantly, this is a lot better way to avoid batch effects when integrating data sets with multiple independent studies than to just be averaging abundances. Now, the next question is, how do we use these health prevalent and health scarce species to predict health or disease? Excuse me. Now, we actually have multiple pages in our paper explaining how we derived and implemented a mathematical formula to predict health and disease, but due to time, I'll briefly describe the main idea in the following. In our view, the most intuitive and straightforward way to estimate whether a microbiome sample is either healthy or non-healthy is to simply find the ratio of the collective abundance of health prevalent species to the collective abundance of health scarce species. And if we put a log on this ratio, and here's the really important point, then any output that is positive means higher collective abundance or abundance of health prevalent species, meaning healthy. But on the other hand, any output that is negative means higher collective abundance of health scarce species, meaning non-healthy. Now, this collective abundance psi is basically a slightly modified version of the geometric mean, which is a reasonable metric to find expected values in microbiome data, given that the relative abundances span several orders of magnitude. After doing the math, you'll be able to find that this version of the mean takes into account the presence or absence of each species within the group, the cumulative relative abundance, and the diversity of those species. Now, moving forward, based on whatever precision cutoffs you select, you can have many combinations of sets of health prevalent and health scarce species. Next, after identifying systematically all possible species sets, we plugged each of them into our formula to test model accuracy. Here, accuracy is basically how accurately we predict healthy subjects to be healthy and non-healthy subjects to be non-healthy. And after looking at all the accuracies, the two sets of microbial species and the parameter tuned model that gave the best accuracy, we end up with what we call the gut microbiome index, which I'll be calling GMHI for short for the rest of this presentation. Here's a table from the main manuscript showing the 50 microbial species we found to best separate healthy from non-healthy. 7 of those species here at the top are the health prevalent, and the remaining 43 are the health scarce species. Now, I know the text in the table is very small, but I like to point out that some of the health prevalent species, again here at the top, are actually marketed probiotics, providing some evidence that these probiotics are indeed associated with health. Okay, so I just went over how we obtained the training data, how we identified microbial species associated with healthy and non-healthy, and how we designed the mathematical formula that can be used as a classifier to distinguish a microbiome sample as either healthy, which is no disease, or non-healthy, which is having a disease. So what do our results look like? First, we differentiate the two major groups, which are the healthy group and the non-healthy group. In this violin plot, we're showing the GMHI distributions of both groups, where the GMHI scores are shown on the y-axis. As I previously mentioned, a positive GMHI means predicted as healthy, and a negative GMHI means predicted as non-healthy. In this way, we get a balanced accuracy of 69.9%, or basically 70%. This balanced accuracy is calculated by taking the proportion of healthy samples we classify correctly and the proportion of non-healthy samples we classify correctly, and just taking the average. The best you can do is 100%, the worst you can do is 50%. The statistical significance of the difference between both distributions is an insanely low p-value, and the effect size of the separation, which we use Cliff's delta to calculate, is 0.56. Briefly, Cliff's delta is a rank-based non-parametric procedure, which goes well with the Mann-Whitney U test. Having a magnitude closer to 1 generally means a better and more consistent separation between the groups. In addition, we broke down the non-healthy group into 12 subclasses. Healthy is still in here, but just crunched on the sides of it. The stars on the top of each of the 12 non-healthy subclasses indicate a significant difference with the corresponding phenotype to healthy. So here, 11 of the 12 diseases show significant separation between against the healthy cohort. One thing I'd like to point out is that we also ran the GMHI algorithm with only metabolic functions, but we weren't able to get good separation between the healthy and non-healthy groups. So although we know microbiome function is important and definitely relative to health, from a classification standpoint, known annotated metabolic functions weren't very helpful in classifying healthy from non-healthy. Anyway, moving on, to truly appreciate and utilize GMHI, we need to consider it as a continuous variable rather than considering as a 1 or 0 or black and white absolutes. Let me explain. And the plot in the middle of the screen, we're binning all 4,347 samples into bins of GMHI. Okay. On the left and right, you have two extremes. In one bin, you have people whose GMHIs are from +4 to +6. And on the bin on the other side, you have people whose GMHIs are from -4 to -6. Now, in between these bins, you have samples binned in a stepwise fashion, leading to both ends. The points on the blue line indicate proportion of samples of each bin that are actually healthy. Points on the orange line indicate proportion of samples of each bin that are actually non-healthy. So the take-home message here is that as your GMHI is more positive or more negative, the higher the confidence we have in the prediction accuracy. The closer your GMHI is to 0, which indicates an equal balance of health prevalent and health scarce species, the less confidence we have in whether you have a disease or not. In this case, probably how your microbiome reflects health status is confounded by non-microbiome factors such as diet, environment, and a bit of genetics. So perhaps this point can be explained further using ordination plots. Here, when we plot all 4,347 samples into a PCOA plot, we see that healthy and non-healthy merge onto each other, showing not so good separation. But what if we only look at the most healthy samples in the healthy group and the most non-healthy samples in the non-healthy group? Okay, we see that if we look at the top 10, 25, 50, and all the way up to top 100, most healthy and most non-healthy samples based upon their GMHIs, we actually get pretty good separation. This shows that GMHI is actually a decent way to stratify healthy and non-healthy groups if we consider the actual index values rather than looking at only positive and negative. But how does GMHI do compared to other metrics? Now, this is not a very straightforward to answer as there is no other microbiome-based metric specifically designed to tell you about health. So just for comparison purposes, we just used what's commonly used in the ecology literature, mainly Shannon diversity, species richness, and 80% abundance coverage, which is basically the minimum number of species you need to compose at least 80% of the total abundance of microbes in a sample. I like to direct your focus to the CLIFS deltas or the effect sizes I had mentioned earlier. As you can see from the small letter Ds in the 4 violin plots, GMHI has the highest magnitude, where again, being closer to 1 means you have a strong separation between healthy and non-healthy. Okay. On each of these scatter plots, we compare GMHI head-to-head with each ecological metric. Each point corresponds to a sample. The x-axis plots each ecological metric, and the y-axis plots GMHI. If you compare the distribution on the x-axis to that of the y-axis, you'll see that the ones on the y-axis, which corresponds to GMHI, always separates healthy from non-healthy better than any other parameter. So up to now, I hope I've convinced you that GMHI can separate healthy from non-healthy and much better so than other indices. But really, the gold standard of any biomarker or health disease indicator is how well it performs on independent validation sets, or in other words, a data set not previously seen during the model training process. Here we show GMHI results on 679 microbiome samples from 9 additional studies. On the left, we show the results when asked to classify between the 2 groups of healthy and non-healthy. As before, the blue and orange distributions are for healthy and non-healthy respectively, and the y-axis shows the GMHI scores. This time we get a balanced accuracy of 73.7% and a CLIFS delta of 0.64. This was really encouraging as we actually did better here than when GMHI was tested on the training set, where accuracy was 69.9% and a CLIFS delta was 0.56. This shows that the overall reproducibility and robustness of GMHI is quite good. Now on the right, we show the GMHI distributions for each class or phenotype of each independent study. As you can see, the 3 healthy cohorts here shown in blue all score mostly positive distributions. And the majority of the non-blue here, non-healthy subclasses score mostly negative distributions. Going from left to right, we're showing the GMHI distributions for ankylosing spondylitis, which is a chronic inflammatory condition of the spinal cord, colorectal adenoma, 3 cohorts of colorectal cancer, Crohn's disease, liver cirrhosis, non-alcoholic fatty liver disease, and rheumatoid arthritis. What's really interesting here is that liver cirrhosis and fatty liver disease, both diseases of the liver, were not seen in the previous training process with 4,300 plus samples, showing that perhaps GMHI can likely be extended beyond the diseases and organ systems it was trained upon. This is really cool because now we can begin to wonder whether we captured elements of a universal gut microbiome disease signature. One thing I'd like to acknowledge is that, as you can see, the tool works around 70% of the time, which means that it's incorrect 30% of the time. So I need to emphasize that despite our efforts apparently moving in the right direction, this is a proof of concept study. In no way am I saying that we should start implementing this health screening tool right now. As with all new things, the tools and techniques and data will get better. Now we have a starting point to go back to the drawing board to not only brainstorm how we can make GMHI more accurate, but also understand why it can be failing in certain circumstances. Next, we show on the right how well the other metrics, Shannon diversity, on the right, Shannon diversity, 80% abundance coverage, and richness do on the validation data sets. Bottom line, not so well compared to GMHI. The main message here is that, and we explain this carefully in our manuscript, GMHI outperforms other metrics by a really wide margin in regards to both accuracy and consistency. And what we found to be encouraging from a validation standpoint is that other people are using our tool too. In this case, a paper that came out in Nature a few weeks ago, studying how diet, socioeconomic factors, environmental metabolites are associated with gut microbiome in a Dutch cohort. As you can see in the highlighted text on the right, the investigators calculated the recently developed gut microbiome health index and identified a significant difference between healthy and unhealthy individuals. Our microbiome signature of health replicated 43 out of the 50 GMHI signals at genus or species level. So this is pleasing to see, as now GMHI is being recognized, used, and tweaked, or what I like to call hacked, for the purposes of independent laboratories. Okay. Now, switching gears a bit, we asked, does GMHI have any relevance to known clinical markers? After searching through all the reported clinical parameters, such as fasting blood glucose, cholesterol, lipid levels from the microbiome samples of our training set, we found that, and this is our last data slide, we found that GMHI is associated with high density lipoprotein cholesterol, or otherwise known as HDLC, or otherwise known as the so-called good cholesterol. If we were to separate people into two groups with positive and negative GMHIs, we found that the two groups had different distributions of HDLC. Now, these are mere statistical associations, and it's kind of hard, at least for me, to come up with a specific hypothesis that we can readily test regarding what we see here. And this is certainly not a strong correlation at all. But nevertheless, we did find it interesting that a health signature of gut microbiome and a marker of cardiovascular health were positively correlated. And it's fun to at least entertain the idea that there could be something going on connecting the gut microbiome to our lipid metabolism, which in turn alters lipoprotein concentration levels. I'd like to mention that scripts and files needed to run GMHI on your own are all available for free on our group's GitHub page, which is provided in the paper. So if you're familiar with Git, please feel free to clone, fork, or send me a pull request if you'd like to play around with it some and contribute. And if working with code is not your thing, then perhaps I can entice the audience to using our tool with the soon to come out website for GMHI, which is being currently built by a really talented undergraduate student in my group. This website is designed for single sample or batch sample runs, and it'll be coming out later in the fall. Now, going back to the image I'd shown earlier. So where are we going with this? Well, on top of a report that provides the high-level taxonomies you have in your gut, or ordination plots describing beta diversities, or in addition to a report on alpha diversity, which I've shown earlier that GMHI does a lot better, or a report on so-called bad species, which any infectious disease or GI doctor can tell you that you should not be having in the first place. or to complement reports on what functions your microbiome has or does not have, or rather providing some ambiguous score that no consumer will ever know how it was conceived, I imagine having a score or index which you know exactly how it was developed and what it's measuring, that you can track at any moment in time, gives you some intuition of whether you're on the right or wrong path, and allows you to compare yourself to others in the population. And even track your GMHI over time to see how well you respond after a dietary or any other lifestyle intervention. Plus, it's kind of fun to quantify and monitor things about yourself. And perhaps even have a friendly competition with friends or family members, like an equivalent of fantasy football, but for your gut microbiome health. So in summary, we designed a stool-based test that distinguishes health from disease just by looking at your gut microbiome. This is really important because if you think about it, cancer, type 2 diabetes, metabolic syndromes, they don't happen overnight. Rather, these diseases take years or even decades to develop inside yourself. If we can look at your gut microbiome and detect something off about you early or prior to the onset of major symptoms, which is when you're really in trouble because it's much harder to reverse the course of disease when it's really progressed, do we then have a potential tool to screen for the pre-disease state? This is a future that I'm personally excited about. To develop our index, we use shotgun stool metagenomes pulled from many independent studies. I want to mention that the fact that we were able to pull this study off with public data, and this clearly shows that there's a lot of value in public data, and we need not only more experts doing data reanalysis, but also more funding for it. GMHI is basically a log ratio formula balancing the collective abundances of two sets of species. One set of species found more frequently in health compared to non-healthy, and another set of species found less frequently in healthy than in non-healthy. GMHI outperforms methods adopted from ecological principles in both training and validation datasets. And last, we believe our study paves the path forward to monitor and predict health, predict human health through non-invasive approaches. Before I end, I'd like to acknowledge my fantastic group at Mayo Clinic, especially Vinod in the middle here, who led this amazing work, and also my generous funding sources, without whom this work would have most likely not have come to fruition. And last, if there's anyone in the audience soon to be looking for a postdoctoral position, and you find what we do is interesting and you like the way we do science, and you want to know what it really means to transform the clinical practice, please reach out. We're always on the lookout for excellent and ambitious talent from all over the world interested in together creating the next innovations in computational precision medicine. That's all for me. Thanks everyone for your time and attention, and I'd be happy to answer any questions or respond to any comments. Thank you so much.